eukaryote and are known as organisms that infect and harm other organisms. Often living in all their hosts, they can be classified into three main groups, which is protozoa, helminths, and ectoparasites, which can cause disease in humans. Protozoa are single-cell organisms that can reproduce in humans. Examples include Plasmodium malariae, which cause malaria infection, and Cryptosporidium, which can cause cryptosporidiosis. These helminths are large multicellular organisms that can usually be seen with the naked eye in their adult stage. They can also be free-living or parasite. Examples include flatworm, trematode, and roundworm. Ectoparasite are small organisms that live on the outside of body, such as tick or mite.
For the material, first Nemipterus japonicus, second Anabas testudinus, third sardine, fourth microscope, fifth cover slip, sixth glass slide, seventh slime water, and lastly forceps.
Together, Nemipterus japonicus, Anabas testinudis, sardine specimens from the suitable aquatic environments. Sampling procedures implement standardized methods to ensure representative samples. Select sampling location considering factors like water depth, temperature, and habitat features. Identification of parasites. Examine each specimen under a microscope. Use forceps to handle specimens carefully. Identify and record the presence of helminth eggs. Identify and record head and tail parasites in sardine. Prepare microscope slides with cover slips for detailed observation. Focus on specific areas of interest such as gills and skins to detect parasites.
For the result about the marine water in Nemicrus spp, we just found the helminth egg. So next about the sardine carrying product, we found the Anisakis sp, so we can see the head and tail.
The discussion of this laboratory, the Nemipterus species from the main water, there have been the nematode life cycle in several stages, including eggs, larvae, and adult worms. While for the sardine from the canine products, there have been the parasite that is known as Anisakis species in fish muscle due to lack of the biosecurity when processing the canned product.
As for conclusion, the presence of parasites such as nematode in aquatic animal, Nemipterus sp and canned sardine indicates that biosecurity need to be improved when processing food. This is to ensure the elimination of parasites and improve product safety. Thank you.
In histology, fungal staining is a crucial method that usually used by the researcher that have a quick identification and management. For the microscopic analysis, lactophenol cotton blue staining method is frequently employed. Then, LPCB are made from three ingredients, which is cotton blue, lactic acid, and lastly is phenol. Okay, all of these three ingredients have their own function. In clinical microbiology lab frequently employ this method to approach the identify filamentous fungus by analyzing their visual.
For the material, first distilled water 200 ml, second cotton blue 0.5 gram, second phenol crystal 200 gram, fourth glycerol 400 ml, and last lactic acid 200 ml.
Preparation of slides. Place a drop of lactophenol cotton blue LPCB stain onto a clean glass slide. For fungal collection, using a sterile needle, pick up a small colony of fungi, spread mycelium by over the LPZP stain on the glass slide. Carefully place a cover slip over the stained fungal material to create a thin layer. Observe the stained fungi under a microscope.
So the diagram 1 show the characteristic of the fungus such as a hype. So we're using the 40 times objective lens.
According to the study, by using lactophenol cotton blue, it can identify the characteristics of fungi. From the result, it showed the hyphae of fungus stained by cotton blue. All these things considered, LPCB staining is a popular and useful technique for observing and identifying fungus.
For the conclusion, the lactophenol cotton blue method is a frequently used staining technique in microbiology, notably for examining fungal structure. It facilitates better visibility and contrast under a microscope, which help with fungal species identification and characterization. The efficacy of the approach is in its capacity to draw attention to significant morphological characteristics, hence aiding impressive fungal taxonomy and studies. Thank you.
White spot syndrome virus is a highly contagious virus that normally affects crustaceans, such as shrimp. It causes white spot on the exoskeleton of infected shrimp that result in high mortality rates and financial loss, especially in shrimp farming. Rapid tests for shrimp WSSV often used to detect the virus to get early identification within a short period.
Okay, for the material, first shrimp sample, second scissors, third sample tube, fourth disposable pestle, fifth disposable dropper, and lastly sample chamber.
Select sample, cut the small piece of shrimp and place it under the or use a disposable to apply mix shrimp using the test kit apply a few drops of shrimp sample to the
The WSSV rapid test show the one pin clip, so that's mean indicate negative.
From the result, WSSV rapid test on shrimp sample showed one pin strip, that means negative result, and shrimp sample are not detected to infect by white spot syndrome disease. This could be more proven by there are no white spot on shrimp carapace, which indicate that shrimp in good conditions and test kit showed accurate result.
For the conclusion, through this laboratory, we knew that rapid test kit used to detect white spot syndrome virus, WSSV on shrimp, because of accuracy result and cost-effective within short time. This detection method can help especially farmers to detect early WSSV in shrimp without takes longer time and even without external laboratories material, which make this method more efficient. Thank you.
NNV, or nervous necrosis virus, is causative agent of viral encephalopathy and retinopathy, otherwise known as a viral nervous necrosis or VNN. Then, a pathogen of viral nervous necrosis (VNS) disease in several important culture economic fish species in fish by provoking a host immune response.
For the material, first fish sample, second scissors, third sample tube, fourth disposable pestle, fifth disposable dropper, and sixth sample chamber, seventh test kit, lastly buffer.
Sample collection. Use scissors to take a sample from the grouper's right eye stalk or the affected area. Place the sample in a tube. Use a disposable pestle to thoroughly mix the fish sample into the tube. Buffer preparation. Follow the kit instruction to prepare the buffer solution. Mix the buffer solution well. Transfer a small sample and few drops of the buffer solution to the test kit's sample chamber. Lay the kit flat and apply the sample buffer mixture to the designated area. Waiting period. Let the kit sit for the specified time for the immunochromatic reaction. After the waiting period, check for the lines on the kit. Double line indicates a positive results, VNN presents. Single line indicates a negative results.
After 50 minutes, VNN test kit show a positive result, means there is VNN in this fish.
Design and establish a rapid diagnostic kit for virus, nervous necrosis or VNN disease using immunochromatography method is helping in determining the existence of the VNN infection in fish. By using the test kit, we can get an instant result to check the fish infection by seeing the line occur on the test kit. In this experiment, it shows that the sample from right eye stalk of the grouper has reacted a positive existence of VNN by showing double line on the test kit.
To conclude, viral nervous necrosis (VNN) is the most important viral disease affecting farm fish in worldwide. One of the species that is easily to be infected is grouper fish. So, with the development of the test kit, it helped to check the VNN at the early stage in a short of a period. In conclusion, the developed VNN test kit provide a sensitive, rapid, and low-cost tool for detection of this virus infection. Thank you.
Gram staining is used in this study to explore the presence and consequence of E. coli and Staphylococcus in aquatic species in specific region. The study goal is to give insight into the environmental and public health concerns connected with their existence, as well as vital information for effective management technique in protecting both ecological balance and human well-being in aquatic environment.
For the material, crystal violet, gram iodine, ethanol 95%, safranin, sterile distilled water, microscope slide, inoculation loops, Bunsen burner, Escherichia coli, Staphylococcus aureus. For the material, crystal violet, gram iodine, ethanol 95%, safranin, sterile distilled water, microscope slide, inoculation loops, Bunsen burner, Escherichia coli, Staphylococcus aureus. For the material, crystal violet, gram iodine, ethanol 95%, safranin, sterile distilled water, microscope slide, inoculation loops, Bunsen burner, Escherichia coli, Staphylococcus aureus. For the material,
E. coli is a gram-negative and has a root-shaped morphology, while Staphylococcus aureus is a gram-positive which has a spherical shape.
Gram staining technique employed in this study provided insights into the presence and consequences of gram-negative Escherichia coli and gram-positive Staphylococcus aureus in aquatic species within specific regions. The varying frequencies of gram-negative Escherichia coli across different aquatic species serve as indicators of fecal contamination, while the identification of gram-positive Staphylococcus aureus raised concerns about potential human health risks. Distribution patterns of this bacteria highlighted distinct ecological dynamics, emphasizing the importance of understanding both gram-negative and gram-positive microbiome communities in aquatic environments. These findings underscore the significance of comprehensive management strategies to address the complexities associated with both types of bacteria and ensuring the protection of ecological balance and human well-being in aquatic ecosystems.
In conclusion, the study's finding on the presence and distribution of Escherichia coli and Staphylococcus aureus in aquatic organisms underscore the importance of ongoing monitoring for potential environmental and public health implications, providing valuable insight for targeted management strategies in aquatic ecosystems. Thank you.